my video response to another CDK007 video in which he talks about human and endogenous retroviruses. It's called Evidence for Evolution Part 3. Now I'm gonna go through his arguments and I'm gonna point out a couple of flaws in his assumptions, and I'm gonna show that basically, in my opinion and based on my arguments, human endogenous retroviruses cannot be used to prove common ancestry with apes. Now, first of all, I'm going to go through CDK007 argument. Now, CDK007's argument is basically this. In both humans and chimps, in the non-coding portions of the genome, you have retrovirus insertions. These are called human endogenous retroviruses. Now, some human endogenous retroviruses, or actually just some retroviruses, are found at exactly the same place in the genome in both humans and chimps. Now, if retroviruses insert into the genome in a completely random manner, you would not find these human endogenous retroviruses at exactly the same place in both humans and chimps, unless there was an infection before the human and chimp split in the phylogenetic tree. For example, let's take the phylogenetic tree. You have the common ancestor for humans and chimps, and you have an infection with a retrovirus, and this retrovirus inserts into the genome of the common ancestor, and the common ancestor, and then you have the split between the humans and chimps, and hence both the humans and chimps inherit this endogenous retrovirus at exactly the same place in the genome. Now, this argument is based on two assumptions. One is implicit and one is explicit. First of all, the explicit assumption. The explicit assumption is that viruses or retroviruses insert into the human genome in a completely random manner. The second assumption is an implicit assumption, and the second assumption is this. The retroviruses are species specific. That is, they only infect one species at a time. They cannot cross-infect two species. Now I'm going to go through and point out the flaws in both of these assumptions. Okay, so anyway, first of all, assumption number one. Assumption number one is that human endogenous retroviruses insert into the genome in a completely random manner. Now, this has been tested extensively in the past couple of years, actually in the past 5 to 10 years, because retroviruses are now used in gene therapy, for example, for such diseases as X-linked immunodeficiency disorder and a couple of others. In these diseases, a virus called murine leukemia virus is used, and which is of the human endogenous retroviruses. So once again, this virus is called murine leukemia virus. For gene therapy, it's very important to study the insertion mechanisms of these retroviruses, because in gene therapy, you basically want to target a specific gene, and so if the retrovirus is inserted in a completely random manner, you would not be able to target this specific gene. So for a couple of viruses, the insertion mechanisms have been studied. These include adenovirus, HIV, murine leukemia virus, and actually for human endogenous retroviruses as well. So I'm going to link up a couple of studies, or I'm going to show a couple of studies on the screen. Okay, so first of all, you have study number one up on the screen, and in this study number one, this talks about the insertion mechanisms of HIV. In this study, you had a culture of T cells, which are a type of immune cells. You actually had a culture which contained 1 million T cells, infected with millions of HIV particles. And after incubating this culture, the researchers could only identify about a couple of thousand insertion points. If the insertion mechanisms of HIV was completely random, you would expect to identify millions of insertion points. Now, HIV isn't that closely related to the human endogenous retroviruses, but another viruses, and this is called murine leukemia virus. Now, I'm going to show another study up on the screen, and this is called Retrovirus Integration Process in the Human Genome. Is it really not random? Which talks about the insertion selection of the murine leukemia virus, and the conclusion of the study is that it's non-random. It's in fact highly specific. So we have two studies, one for HIV and one for the murine leukemia virus, showing that the integration site selection is not random, and in fact it chooses specific sites to insert into. Both murine leukemia virus and the HIV choose specific sites in the genome to insert into. Okay, and then on the screen I have a third study, which deals specifically with human endogenous retroviruses, and the site selection for human endogenous retroviruses is highly specific. So now we have three studies, one dealing with HIV, the other with murine leukemia virus, and lastly the third study is dealing specifically with human endogenous retroviruses, proving that the human endogenous retroviruses integrate into the human genome in a highly specific manner and not randomly. And now I'm going to go on to the second assumption and just prove to show how it was wrong as well. Okay, the second assumption is that the retroviruses are species specific, that is, they do not cross-infect species. Now, we know this is wrong because several retroviruses, for example, adenovirus, HIV, MLV, which is the murine leukemia virus, mouse mammary tumor virus, all of these are not species specific, but in fact both humans and apes, or actually humans and other animals. For example, HIV infects both humans and chimps. The adenovirus infects both humans and chimps. The human T-cell lymphotrophic virus infects both humans and I forgot which other animals. Anyway, none of these retroviruses, or very few of these retroviruses, are species specific. Now, why is this important? And why is it important that the integration site selection for retroviruses are highly specific? I'm going to explain both of these in a second. Okay, now imagine this scenario. You have a human and a chimp and a retrovirus. Now we know that this retrovirus is not species specific, so it will infect separately both the human and the chimp. And we know that this retrovirus is highly specific in choosing which area of the genome to insert to. So in a separate infection of a chimp and a human, this retrovirus will choose the same area of the genome. So separate infections in humans and chimps can account for retroviruses being found in the same place in humans and chimps. In fact, if these two assumptions are correct, number one, that retroviruses are not species specific, and number two, that retroviruses are highly specific in choosing the area of the genome in which they integrate, it is inevitable that some retroviruses will be found at exactly the same place in both humans and chimps. Now the next question is, if this scenario is correct, how many endogenous retroviruses are expected to be shared between humans and chimps? Well, currently there are 98,000 human endogenous retroviruses in the human genome. Out of the 98,000, there are 16 shared between humans and chimps. Now, since we already know that this scenario is correct, and that retroviruses are not species specific, and that retroviruses are highly specific in the area of the genome they insert into, it is inevitable that at least 20, 40, 60, maybe 100 endogenous retroviruses will be found at exactly the same place in humans and chimps from separate infections. Hence, human endogenous retroviruses prove absolutely nothing about common ancestry.